Welcome to the next module. In this module, we will be discussing the biotechnology field. So as you can see on the screen also, I have this presentation that I've designed for you so that you can at least understand how you can complete your PhD specifically in the area of biotechnology. Biotechnology is a field of multiple different disciplines. You have microbiology, you have cell biology, you have immunology, biochemistry, and all. All these fields, they are gathered together, and you can say it's a life sciences. That is the major domain, and this particular course, entire course, will help you understand that in life sciences, if you want to do PhD, that what are the different aspects for completing the PhD. We have already, in the previous module, we have already discussed the mindset, the PhD mindset. Once you have that mindset, next thing you have to complete is finding out the domain, right? So I'll help you in finding out that particular domain. I'll guide you through that. What are the different domains available? How you can find out that particular subdomain which is close to your area of expertise, which matches your personality, and etc. So you can see over here, I have mentioned the topic as choosing biotechnology with intent. So you need to have that mindset that you need to complete biotechnology. You have to complete the PhD in biotechnology. So there are mainly three factors based on which students they select their field. One is peer influence. Most of the time what you'll see is you select a particular area that is usually dependent upon maybe your friend or maybe your relative or maybe your known person have selected that particular field, and based on his or her experience, you jump onto that area and you feel connected to that particular area. Maybe your parents, they have some experience in biotechnology. So that peer influence is always going to be there, and based on that, you will be selecting the area. But you have to be careful, right? You have to find out what will be your own core interest. Let's say I'm interested in microbiology, but my friends are suggesting biotechnology again and again. So you need to understand that you cannot be influenced by those factors. You have to listen to your inner voice and also your inner interest that specifically control the outcome of that selecting a particular field. So you should not be selecting the field just based on what people are saying or what trend is going on. Maybe right now trend is related to artificial intelligence in biology, and a trend, they can change, right? But your core interest, your core focus is never going to change. So your PhD is going to stick with you for your entire career. So you need to be very, very careful selecting that area, right? Next is supervisor availability is another factor. Sometimes you're working in the same university and you're connected to other universities, and you find people, of your same mindset. You find people who are connected when you were doing PG, UG, and you want to select them as supervisor, and they are available. Based on that, you can also, have that decision done. Most of the time, what I've seen is selecting supervisor as another, challenge that people face. So we'll be discussing on that in detail, but you need to find out a supervisor who is having that availability, who is having the seat, matches in your area of interest, and then you have to start your PhD. So doing research before research for PhD is really important. You cannot be just jumping directly on PhD and then suffer a lot, later regret that what you have done. So that is really, really important. But anyways, if you have not done that exercise, because these things, they are not there, nobody's going to guide you through that, right? So it's completely fine. I'll help you with the entire process. So there is no need for you to worry. But one thing you need to ensure that supervisor, the guide, the PhD fellow, who is going to be your professor, is available, is having those seats, and then you can, easily proceed. Next is trending keywords or trends. For example, sometimes people think that AI in biotechnology, computational biology, or maybe vaccines, infections, viral infections, something like that is always there. Some trend is going to be there, and people, I don't think that's a correct idea for doing research. They focus on those trends and then they try to select the area. So in my opinion, I don't think that's a good choice. I would say you need to be careful while you're selecting your particular area. right? So do not focus on trendy words. Focus on the problem. Focus on what really you want to solve, what really challenges you. If cancer is the area that you think it excites you and you are focused and you are really motivated to find out the cure, please go ahead and do research in cancer. But you should not be selecting cancer based on other people are saying. So many people are working on cancer. When you are talking in society, you are talking about cancer, everybody is getting excited. So just based on that, you should not be selecting. You can be selecting an area which is really unique. Nobody's working on that area. Nobody's working on that particular problem. That is something is going to make you special in that area. In that field even, right? So biotechnology will have multiple areas. You will select the particular area that you want to select for your PhD based on various factors. Some of the factors that I've discussed over here, hopefully now you have a better clarity on those. Next, let's talk about, early confusion and what is the impact of that confusion later on. So let's say if you're confused and you're randomly selecting that particular area, one thing is going to happen definitely is wasted years. You will be spending years on working on a problem which you really don't like, and you don't want to do that, right? You don't want to select an area which is not of your interest. Next is, misaligned PhDs. Your interest is going somewhere else. You're working in an area that is not of your interest, so it will be really, really difficult for you. And let me tell you honestly, it is not going to be like that. You will not select directly an area. You will be really, really fortunate, and maybe you're doing a lot of research at UG and PG level, and then you will be knowing that this is my area. Sometimes it happens that you get a topic which is not really aligned to your interest, but try to align that. And how to do that, then we can also discuss later on that how you can align your PhD in a particular area that is of your interest, right? But make sure you select the area based on your interest initially, so there is good alignment between your PhD and the topic that you've given for your PhD. And what is your interest. All those things should be aligned. Burnout during research is very, very common, because if you are working on something that you are not really interested in, PhD is something you have to go really, really deep, right? You have to go and do research on that particular area for years. So you should not be working in an area which you don't like. So burnout is going to happen very, very quickly. So if you have correct mindset, you're going to select a particular area which is fun to you, which is really exciting to you. And if you jump onto that area, you will feel energetic every time you work on that particular project. So that is something you need to be very, very careful. Now let's talk about major subfields in biotechnology. As you can see over here, you have different areas. So whenever you select microbiology, you have multiple areas, and you will find supervisors in those areas. You can have microbiology, so infectious diseases. I've kept it on the top because that is my area. That is my area of research. I'm a little bit biased on that, right? So microbiology and infectious diseases, most of my PhD students, they are working in this particular area, and I just want to work in that particular area, not deviate too much in and around it. Next is cellular and molecular and cell biology is another area where you understand cellular functions in microbiology. Okay, so let me tell you what happens in microbiology and infectious diseases, right? And then let me explain all these areas with some examples so that you can understand what I'm talking about. One is microbiology and infectious diseases is the area where you see the infections that are going on with animals, with humans. You have viral infections, you have bacterial infections, you have antimicrobial resistance, all those and there are so many other things that can come under infectious diseases and microbiology. General microbiology, you can have industrial microbiology, you can have food microbiology. So many different areas can be there in microbiology, right? Next is molecular and cell biology. In this, you will be understanding cellular functions and understand maybe some diseases that are associated with cell biology. Let's say cancer. You may be also working in the area of cellular and molecular biology, considering diseases like Parkinson, neurological diseases. All that comes under molecular and cell biology, where you are trying to understand cellular functions. And cellular function most of the time here is focused on eukaryotic cells, right? Okay, so that can be the area. And molecular biology is something that you are working and understanding the functions of DNA, RNA, proteins, and all. So that comes under that area. Genetics and genomics, right? You need to understand different genomic parameters of particular organism or species. Let's say you want to understand the genetic makeup. What is the impact of that genetic makeup on the phenotype? And there are so many genetic diseases that are there. That is the area. Genomics, you want to understand the genome profile right now. What is also happening, which is another important area of research, people trying to understand that how our genetic makeup influence our our life, susceptibility to diseases, aging, all those things people are digging deep into. Right now, what we are happening is we are getting lot of powerful sequencing machines, right? So a lot of people, they are working in the area of sequencing and understanding the sequence. They're doing PhD in that area, right? And further, genomics becomes their core area of research. So they want to understand, how sequence of the particular gene helps understand our overall health. So that is really, really important. I think that area is going to, increase in future. A lot of people are going to work in that particular area because it's really exciting. Right now, we don't have instrumentation. You don't have facility to do sequencing quite often because it's really expensive. So if we can find out something that can help us getting sequence at low cost and also very, very fast, we should be able to get a lot of interesting research out from that area. Next is immunology and inflammation. Another area that people work on, that how our immune system fights against the infection. That can be against infection, that can be against tissue injury, that can be against our own antigens. That's immunology. We all know that what immunology is, right? Bioinformatics and computational biology. Right now, what is happening that people are very, very inclined towards artificial intelligence, and that is very close to bioinformatics and computational biology. So people are working in the area of computational biology and bioinformatics because it's very close to artificial intelligence, right? Where we try to understand the structure, function of cellular genes, proteins using computational power. So there are tools available. One of the important aspect is you want to understand the sequence, you want to compare the sequence and all. So that comes under the bioinformatics. But my recommendation usually is if you can combine experimental research with bioinformatics, that becomes really something really important. For example, you are just someone who can understand the sequence and analyze the sequence. That is fine, but if you also know how to do sequencing experimentally, you become really powerful. So in your PhD, you should mix up experimental as well as computational biology together. And that is what we do in our lab also. We combine computational biology with experimental results, and we are doing good so far, right? So that is really important. Next is structural biology and biophysics. So a lot of work is going on how biomolecules, they behave, how interaction happens at molecular level, how they are interacting, combining, how they are getting dissociated, what speed they are working in, how they move from one place to another one. So that's the biophysics. And my postdoc was in biophysics, so I can tell you this is something is really, really unique area. If you have some interest in quantitative sciences, you're good in mathematics, you're good in physics, you should be heading towards biophysics. That is the area that you should be working on. That's exactly at the interface between physics and biology. That is your biophysics over here. And structural biology is basically a lot of people, I'm sure you have seen that they solve problems associated with structure of various molecules, how they interact. That mostly is the structural biology, right? Industrial environmental biotechnology. Now you have a lot of industrial applications, like you have a lot of enzymes. You can apply those enzymes and solve a lot of challenges. There are dyes in the industrial waste. You have wastewater that can be treated. That can come under industrial microbiology or industrial biotechnology, right? And environmental biotechnology. That is something really important area. People are working in this particular area, and they are getting a lot of impactful papers out, because a lot of papers you'll see in the environmental sciences are really, really good journals. So a lot of people are they are interested in getting people who are trained in environmental biotechnology, environmental microbiology, because there are so many challenges associated with the environment, right? So this also becomes a very, very attractive area of research in the area of biotechnology. Next is drug discovery research. That is also a kind of, you'll say, interface between pharmacy and biotechnology, where you understand the mechanism of drug. Also at the same time, you can try to find out new molecules that can work against a particular disease. So you'll see most of the pharmacy people, they work in this particular area. But as a biotechnology, you can also as a biotechnologist, you can also work in that particular area. So these are some of the areas that I mentioned. If you have any doubt, if you want to know about some different areas, then you can just contact me. You can contact me, you can email me, you go to the ResearchGoal website, you can just shoot an email. That email will immediately come to me. And if you have taken up this particular course, please mention the ID, student ID, so it helps me answer the queries a little bit faster. Okay. So next is after this, research track within each field. This is really important. within one particular field you will have different tracks even, right? So you have biotechnology, then you have different areas down there, and then you have different tracks in between. So we'll start with one discovery driven research. So what you can do sometime is you want to identify a particular mechanism, you want to identify a particular pathway, hypothesis, you want to discover something, right? So that is discovery associated research that you will be doing. Next is method driven research. You want to standardize a particular method that will detect something. So you can develop a tool, maybe an online tool that can do some kind of research. You can develop assays, detection assays. You want to detect COVID antigen, you want to detect something else, you want to detect viral protein, bacterial proteins. So that's assay driven research. Techniques also, you want to create a new technique that can do something new. Like there are assays available, ELISA is available, you want to develop something else, PCR is available, right? Those are the output of the research. I think this research gives you a lot of money. It's more like applied research. The first one, discovery driven research, is basic, but if you can get molecules out, it becomes really good for your research funding. So both of them are really good. I hope I was able to explain those things. Next is data-driven research, where you try to collect the data, you try to analyze the data, and you try to get insight. You can do big data analysis, modeling, prediction. That is more like you have computational biology, and then you're doing all those things together. Next is application-driven research, which is very, very close to the method-driven research. In application-driven research, you are focused on the final product. You are focused on creating something, not just assay, not just method. You are focused on a final finished product, and right now, a lot of people are working in that particular area. They want to have the PhD focused on a particular product and maybe diagnostic or maybe therapies. So final therapy that they want to create during their PhD. So you can see that when you're designing your entire PhD synopsis or project, you should be taking these things into account. You cannot be just mechanism driven research. You cannot be just method driven research. If you want to do that, then that should be your area of interest, right? You should be focused on those areas, and you should be really interested in those. So this is how you can also select and talk to your supervisor that I'm interested in this one. Can you design something in and around in this, focused on your area, obviously. And let's say maybe I'm interested in assay development. So you can work on specific assay development. And that is really, really good. If you want to move to industry, that is something is really going to help you. This is not going to help you mechanism and pathways dependent research. That will help you in moving into academia, right? So you can understand if your research is more towards the mechanism based, the academics is really good for your future. If it is more toward product based, industry will be really, really good for you. But although universities also, they are interested in getting people who can do applied research, but you can understand you have more opportunities on that side, industry side, if you do the method driven research and application driven research, right? Okay, so next is matching personality with the research domain is really, really important, that you need to match your own personality with the research domain that you're working in. So you can have, first is analytical. If you want to be more, if you are analytical in your head, you are more like trying to solve a particular challenge. You can select molecular biology, structural biology, as I told you, right? Structural biology, all those requires a lot, and biophysics also requires a lot of quantitative kind of nature. So if you are in that domain, if you are interested in that particular domain, and you should be going into that area, like in analytical research. Next is exploratory. So that is basic sciences, mechanism, as I've already told you, just trying to find out something new, trying to find out something really, really new. Nobody known about that particular organism. Maybe you're just trying to find out new organism, which is before it was not known, right? So that is something really basic. You're just doing research, trying to find out something really new. So that is exploratory for you. Next is computational mindset. If you have computational mindset, you like working on computers, you are not that much of a lab kind of a person. So computers, if you like computers, bioinformatics and AI driven biology is really attractive choice for you. So based on that, you can find out people who are working in that area. Next is social impact driven research. That area is, I think, is really critical. Not many people, not many biotechnologists focus in a sense that they want to do research focused on social aspects. Doing something is really good for public. So that is the area you can also focus on, like clinical research, public health research, translational research. All these things you can do. Like various hospitals are available. You can go and enroll into that hospital for your research work, and you can also enroll into a university, because most of the time hospital is associated with the university, and you can do that kind of research, right? So that will be the social impact driven research. Okay, so these are some of the important aspects that I've covered so far, focused on biotechnology as the domain, and I'll tell you everything in and around it. So it's better for you to prepare in advance, do your homework, pen it down, write it down somewhere, that what are the areas that you want to select on. Select the major area, focused, narrow it down, go more detailed, whether you want to do method driven research, whether you want to product driven research, and based on that you can formulate your proposal. That will help you get into the PhD program very, very easily. If you are doing UG research, you can also do same thing so that it aligns with your postdoc and not postdoc, with your PG and your PhD. It's really, really important, because many times that supervisor, they're going to ask what kind of project you have done, what was the technique you have followed. So if that is aligned with the supervisor, obviously directly you're gonna get that benefit, and you will be easily selected, right? So I hope all the things associated with the domain, subdomains, and also research pathway that we have covered in this particular module, and that will help you aligning your thought process to your future PhD lab, right? We'll be covering more things like that, and complete all those modules, and let's see by the end of this particular course if you have better clarity on your PhD. All right, if you have any questions, please do let me know. You can just shoot an email and I'll try to answer your queries as soon as possible. All right, let's move on to the next one.